up here. I know I can't draw. I made this big so we could fit everything in. You guys probably are much better. Start out here, all right? Start out here. What is that? This is the big picture, all right? I'm out here in my finger. This is a receptor, all right? And if I told you it was a light touch receptor, you'd probably tell me it's what? It's a mechanoreceptor. So I am taking all this fun PowerPoint text notes and I'm putting it in a picture, and this is how I write my exams, all right? So here's a light touch receptor, mechanoreceptor. It's out here in my hand. Do they generate action potentials or graded potentials?
Graded.
Graded potentials. These are all things we talked about today, but you were listening without your coffee and you didn't really understand what was going on. But at receptors, we do graded potentials, which in my idea is the first domino. Do you understand that? The first domino is knocked over here, okay? And we're going to carry this signal, and I already drew this for you because Jason asked me to earlier.
Wait.
Well, we kind of spoiled the surprise because I drew this earlier and labeled it for you. This is a sensory neuron. Do you understand that? This is a sensory neuron because we are carrying the signal toward the CNS. Structurally, this is called what? This is the hard part now.
Not the axon.
Good. I like that. I like it called the ALD, which is axon-like dendrite. It is a dendrite. But geez, it sure looks like an axon.
Will we find ALD anywhere else besides this class?
No.
Okay.
Mm-hmm.
I won't Google it.
Okay.
You can Google it. I mean, it's accepted as
Oh,
a
is it?
theory. It's just
Okay.
Marev doesn't teach it this way.
That's fine.
Yeah. But no, it's a theory. I just made it up. So, it's myelinated, which is odd because Marev says dendrites are myelinated, but they are, clearly. All right. In the way you're teaching this, I didn't mean to be funny. That came off kind of nasty, but I didn't mean it that
No,
way.
I was in a class where a guy he said this is this, but I like to call them these.
Right.
You don't have to use that.
I made it up.
So,
No, this is a accepted theory. You know, Marev talks about, dendrites not being she would call this an axon. But by definition, axons carry the signal away from the cell body, so I've never understood how she can present it that way. So, that's okay. Dendrites are carrying the signal toward the cell body, and it is myelinated. Can you take an area here? What would this area be called?
A node of
Gap.
maybe.
Oops, not.
It is a gap.
It's a node, right?
Something.
Right, gap in the model. It's called a node. All right, fine. They're typically called nodes. But geez, what do we have at nodes that you need to know about? Located at the node, we would have a voltage-gated sodium channel, a voltage-gated potassium channel, passive channels, and what else? Our good friend,
Pump.
the pump. So wherever you have this gap in the myelin, you have these four things. And this is how we're able to propagate action potential and move the dominoes all the way down to here. If I expanded this section up here, and we called that the dendrite, what could you tell me about the ions on the inside versus the outside? High potassium. inside, high sodium outside, just like every neuron in the body have resting membrane potential. Questions on that? Okay. So we continue down the nodes with our action potential. And we get to the cell body. Which is outside the CNS, so it must be in a structure called a what?
Ganglia.
Ganglion. And this one actually is the dorsal root ganglion. There's the axon. And the ATE. Axonal terminal ending. Would be a good idea to magnify this. This little area here you see what I did? I took this area here, which is a synapse between the sensory neuron and an interneuron. And I magnified it. You guys know how this works. I'm just making a point. What do you find? If this is the ATE here, coming down this way, what would you find inside these circles?
Neurotransmitters.
Neurotransmitters, all right. Neurotransmitters, acetylcholine for our purposes now. And when the signal, the action potential got down here, what channels would open right here that are stimulated to open by the action potential? Voltage-gated what? Oh, calcium. Good. Calcium rushes in. We get fusion of the synaptic vesicles. The chemical reaches into the cleft, binds to the postsynaptic membrane, and now, Casey, we can actually ramp it up a little bit because we now know that the postsynaptic membrane of the next neuron, those are chemically gated sodium channels. Chemically gated sodium channels, and sodium rushes in.
But it doesn't start the thing like on a muscle or a circle on it. It goes from a chemically gated to a voltage gated.
Here,
And that cleft does the same thing.
what does that create?
Action potential. But deep hole.
Yeah. What does it create when the first domino opens on an adjacent channel?
Graded potential.
Gradient potential. It's a gradient potential. It's the first domino, which will then kick over the next channel, which is a voltage-gated sodium channel, and away we go. So we click over the dominoes, and now, are we sending the signal down the interneuron? Choo, 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 till we get to our motor neuron, which is another synapse that looks just like this. Now we start to send the signal out. The motor neuron that is myelinated, and it has the nodes. And if you bring the nodes down, same information is found there, and we're propagating the signal down the motor neuron. to the end of the motor neuron. Can you draw that? Man, this was exam number 3. Yeah. Exam 3. I bet you guys do this in your sleep. This is the end, the ATE, the end of the motor neuron, acetylcholine, synaptic cleft, sarcolemma, T-tubule. You see where I'm going with this? That's how we get it all together. This is the cycle of life. When this fuses and is released by exocytosis, neurotransmitter in here, what's it bind to? Chemically gated sodium channel, beautiful. On the postsynaptic membrane. It's what we just kept going through. We're just moving to a new location. Chemically gated sodium channel. Does that generate an action potential or graded potential, Jason?
Action. What? Oh.
I picked on you because we had it backwards before. A neurotransmitter at a chemically gated channel is the first domino, and that is a graded, which eventually will become a what, Jason? Action. All right. This is graded, and the next domino will kick over, voltage gated, voltage gated, voltage gated, and all of a sudden we're headed down the T-tubule just for kicks. That action potential, as it moves down, can muscle cells generate action potentials? Apparently. It's actually in your notes today. Things that can generate action potential, sarcolemmas. So here it was. Goes down there. Anybody remember what kind of channel that is?
Voltage gated calcium.
Voltage-gated calcium. Calcium floods down to our good friend troponin. Stop there. You don't have to know that for the next test. But you get the idea, is that this, on our third exam, is a lot bigger than just muscle contraction. It's now, can you take a signal from right here and run it down your arm, into the spinal cord, and back, and name how it all works? With the channels, and the locations, and the nodes, and the sensories, and the motors, and that's it. And then also add in depolarization, repolarization, millivolt potential change, which I talked about today. That's neurophysiology. Next week, to get to the heart. This is where I was talking about, I think. The skeletal muscle, if that is the effector, it takes one motor neuron to get to it. Y'all with me on that? What I need to add in is smooth muscle. You got smooth and cardiac. Because if this is actually smooth muscle out here, as the effector, would there be smooth muscle out here?
Yes.
Yeah, blood vessels, erector pili muscle, all right, they'd be out here. It takes actually 2 neurons to get there. So we start in here, we go to here, we synapse again. And it takes 2 neurons to get out. Skeletal muscle only takes 1. If we synapse here with another neuron, and that's the cell body, that must be in a what? Ganglia. Good. And there are 3 ganglia I'll introduce to you on Monday.